Hello everyone, this is the presentation of our research. As you can see, our topic is behavior of planarian individuals under the stimulation of light sources. First, we will introduce how to feed planarians. Secondly, we use pretest to confirm that our experimental device is highly trusted. Lastly, we will introduce our formal experiment and our interpretation to these experiments. Look at this picture. This is the device that we use to feed planarians. First, we install a water filter. Secondly, we feed them in a dark room. Thirdly, we put few stones in order that planarians can cling to them. Lastly, we feed them with an egg without egg white on Friday morning and refresh the water in the afternoon. Next, we are going to talk about our pretest. This is environment control. Planarians must be starved for 2 days before an experiment, and the experiments are performed in a dark room that is 20 degrees Celsius. As for our purpose, we did this experiment in order to confirm that our experimental device is highly trusted because planarians' negative phototaxis is a well-known condition. This is the experiment device. We put planarians into the bright district. 30 minutes later, we count the number of planarians in the dark district. At first, in every group, all planarians were in the bright district. 30 minutes later, almost all planarians were in dark district. However, we found that one planarian was still in the bright district. After our careful inspection, we found that planarian was headless. Now, we are going to talk about our hypothesis based on the previous references. According to previous references, it shows that planarian's body can also sense light. Therefore, we think that planarians should minimize the light receiving area after being illuminated, and the blue light would have the most reduction.
Next, we are going to talk about our formal experiment. We use 3 Petri dishes and 3 colors of LED light as our materials. Then we put all of this into a constant temperature cabinet that is maintained at 20 degrees Celsius in order not to let the temperature become a variation of our experiment. First, we put 3 planarians into each Petri dish, take pictures and record. After 4 hours of exposure to the light, we remove them and record it again. Secondly, we analyze the pictures to compare the information we need, that is, individual distances and the changes in the body length. Our analysis points are shown in the PowerPoint. We calculate the individual distances by before minus after, and percentage of the remaining body length by after divide before. In this form, we can find that the individual distances of planarians are similar in different lights. In this form, we first average the data of 6 groups that illuminate the same light. We can find that blue light makes the individual distances of planarians the shortest. In this form, we compare the data of 6 groups that receive different light. We can find that blue light also makes the body length of planarians change the most.
As the data shown in the PowerPoint, In Form A, we can find that the influence of the red light is approximately the same as that of the green light, while the illumination of the blue light makes planarians gather the most obviously. In Form B, it's a little bit different. We find that the influence of the green light is approximately the same as that of the blue light, while the illumination of the red light makes planarians remain with the longest body length.
Here we list the influence level of different light sources. The most special point is that the influence level of illumination under the green light is low in individual distances, but high in body length variation. Our prospects mainly emphasize the density of the planarians, and we are doing this now. The others are the illumination of different light sources and a deeper exploration in green light.
This is the end. Thanks for your listening.